In the introduction lecture for this course, I showed a slide that showed all the levels of biological organization. The lowest level of biological organization is the molecule, and that's why we spent time in chapters 2 and 3 learning about molecules. Well, now we're going to combine those molecules into living cells. So if you remember from that slide, the first level was molecules, the next level was organelles. Organelles are basically organs inside of a cell, and not all cells have them. Some cells do, some cells don't. But our molecules are organized into cells and organelles if they're present. So we're just sort of working our way up that biological hierarchy. And we're going to spend most of our time, the rest of the semester, looking at cellular processes. We'll look at the structure of the cell. We'll look at processes that the cell performs. We'll look at how DNA is used in a cell. And so that is the focus of this chapter, is just kind of learning about the roadmap for the cell. What structures are present in the cell and what do they do? So just like our other chapters, we've got our learning objectives. There's a long list here of things that you need to know. You definitely need to know the cell theory. That is not in your textbook. You're only going to find that in the lecture. Another big thing that you're going to have to do at the end of this chapter is be able to compare and contrast a prokaryotic cell and the types of organisms that are prokaryotic with a eukaryotic cell and the types of organisms that are eukaryotic. And then once we have those eukaryotic cells down, you'll need to know the parts of the cell. So this is basically cellular anatomy. What are the structures that build a cell? And then we're going to introduce a little bit of evolution here. We're going to talk about the origin of eukaryotic cells and how they actually came about. So we started off our biology course with chemistry. Now, I've already mentioned a little bit about why we study chemistry, because chemistry is sort of the lowest level of organization. All of our cells are made up of molecules. And in chapter 3, we spent way too much time on some of these biological molecules, and some of you were probably hoping you never had to think about them again. But they're all really important because they all play really important roles in the structure and function of our cells. So just a quick recap from chapter 3. Lipids are important because they enclose our organelles and our cells. They make up the plasma membrane that surrounds our cells. They make up membranes inside our cells that surround our organelles. Carbohydrates are really important because they provide energy. And we'll spend a whole 2 chapters on energy, actually, learning how cells bring in and convert energy into usable forms and then actually use energy. Carbohydrates also provide structure for some of our cells. Proteins are important because they carry out all of the molecular work that happens in a cell. They are the little worker bees that go in there and take the information from DNA and put it into practice. We won't talk about proteins too much in this chapter. Mostly we're going to talk about lipids and carbohydrates, but proteins are definitely important later. And then, of course, nucleic acids. We won't talk about those too much, but their structures are really important in running the cell, in providing the instructions for our cell to function properly. So those molecules there, they really are the foundation of all of the activity that happens in our cells. Once we get from the molecular level, we start to combine all of those molecules together to produce specific sort of organized structures in the cells, specific structures within our organelles, all with a very specific function. So if you remember from chapter 2 and 3, we talked a little bit about emergent properties, where you organize things in a specific way that gives it a very important function. We also talked about structure and function, where the structure leads directly into the function. So those molecules that we were looking at in chapters 2 and 3 are really important because they are foundations on which the cell is built. And since we're biology, we're studying life, we have to start with that smallest unit of living, which are the cells. And those atoms and molecules that we studied in chapters 2 and 3 make up those cells. So let's do a really short history lesson. If I ever give you a history lesson or talk about specific people in the lectures, you do need to know them. Would love to go into a lot more history. I find the history of biology really fascinating. We just don't have time. But if I do talk about specific people, it's because they were really important, and I do want you to know who they are. So there's a few pictures here of some of the individuals that did microscope work and early cell work. We are going to start with the invention of microscopes. So microscopes were invented in 1590. So that's like 425-ish, bad math, years ago. So microscopes have been around for a long time. They've come a long, long way. You can see a picture down there in the corner. That's actually a microscope down there. You would put a little droplet of water on the tip of that point there and look at it through a piece of glass, a curved piece of glass, and that allowed magnification to happen. The first cells weren't observed for a little while. About 350 years ago, Robert Hooke observed the first living cells. I'm sorry, not the first living cells, the first cells. He actually observed cork tissue. So think of a cork that you would find in a wine bottle. That's naturally occurring plant material. And he called them cells, these little regions here, because they reminded him of prison cells, which is a little sad, but that's why we call them cells, because Robert Hooke first observed these little chambers inside cork tissue. Not long after that, Anton von Leeuwenhoek modified the magnifying glass and was actually able to, for the first time, see living organisms under a microscope. So this is Anton von Leeuwenhoek there. This microscope down the bottom is actually his microscope, and he noticed little things swimming around in pond water. So he didn't know what they were. Of course, no one had ever seen them before. So he just basically called them little animals. There were little animals swimming around in the pond water. But that's relevant to us because that's the first time anybody saw living cells. So at this point, we now have a microscopic world. And we're starting to notice that things are made up of these little tiny structures. They're either entire animals, in air quotes, like he was seeing in the pond water, or they are part of a larger structure like we see in plants and now that we know make up animals. So continuing on with our history, we had a couple of cell biologists who took Leeuwenhoek's and others' research and sort of put together the idea that plants and animals are actually made up of cells. So our microscope work continued, and in the 1830s, a botanist and a zoologist, Matthias Schleiden and Theodor Schwann, came up with this sort of grand new idea that plants and animal cells, not plant and animal cells, plants and animals are made up of cells. So they are a compilation of a whole bunch of cells with all different functions that are all working together to produce the organism and all of its glory. In the 1850s, we have a few other scientists, Virchow, Remak, and Kolliker. They stated that all cells were made from pre-existing cells. Now, this is really important. This is basically them saying that reproduction happens. Organisms don't just poof out of nothing. They have to actually come from a pre-existing cell. And that was pretty groundbreaking at that point because most people didn't even know what a cell was. The idea of something microscopic was kind of mind-blowing and unbelievable. And so this idea that one cell leads to another is really pretty important. In 1859, Louis Pasteur, who has been an important figure in your life, even if you don't know about it, basically refuted this idea of spontaneous generation. He supported Virchow and others' idea that cells must come from pre-existing cells. They don't come out of nothing. So Louis Pasteur is the man that gave us pasteurization, and he did a lot of work with microorganisms, and he basically discovered that if you heat up liquid, you can kill the microorganisms, and if you do it correctly, you don't ruin the liquid itself. So we pasteurize milk and other liquids that we consume to get rid of the bacteria without essentially cooking the product. He came up with all kinds of other things. He's a really smart man that really did a lot of work in microbiology. But without going into too much detail of his experiment, he basically showed that cells don't just spontaneously generate. And he had this really cool apparatus called the swan neck flask. And basically he put some liquid in there, non-sterile liquid, which means there were probably things growing in it. And then he heated up the neck of the flask. This basically killed any living organisms that were living in that flask. And then he heated up the solution. So he sterilized the solution, killed all living organisms. Well, when he did that, nothing grew in there. So basically, by sterilizing this swan neck flask, he was getting rid of all microorganisms. And then nothing could grow in that liquid because nothing could get there. That's the whole point of this swan neck here. All of the living organisms sort of remain in the front end. But the important thing was that for many years, nothing could grow in that liquid. And he was able to establish that cells must be present for new cells to appear. He showed the opposite as well, that if that liquid had contact with the outside non-sterile air, you would get microorganisms growing in there. So if a living cell could enter in, you got more cells. If the living cell could not, you didn't get cells. So that was really, really important. So if we take all of this information, Schleiden, Schwann, Virchow, Pasteur, we get what we call the cell theory. And the cell theory helps us to make a definition for what living means. With the cell theory, we get the idea that all organisms are made up of one or more cells. So anything smaller than a cell, an organelle, a virus, are not considered living. Viruses are their own kind of special though. They have some characteristics of living cells, but they don't do all the things that cells do. So all organisms, if you're gonna be a living organism, you have to be made up of at least one cell. Cells are the fundamental and structural unit of life. What that means is that there's nothing lower than a cell for a living organism. So 1 and 2 kind of go together. All organisms are at least one cell. A cell is the smallest structure that we would call living. And then last, and this comes from Virchow and Pasteur, all cells must come from a pre-existing cell. So this means, of course, that there is reproduction. So all cells reproduce. It also means that even multicellular organisms somehow have to pass on their information in a cell. So that's our classic cell theory. I want you to know these 3 tenets of the cell theory. Our modern cell theory is a little bit more complex. We define a few more things that make something a cell, but they're all kind of based in these 3 tenets. So I want you to know those things. If you are interested in this cell theory and a little bit of this history, there are some articles in the Chapter 4 folder in the weekly course content in Blackboard. So we've talked about what or where, not what or where. We talked about what defines life, basically. We stated our cell theory. Now we're going to talk about what it means to be a cell. So there are 2 types of cells. We've got prokaryotic cells, and we've got eukaryotic cells. So when I ask you what are the 2 main types of cells, your response are, one is prokaryotic and the other is eukaryotic. We'll talk about some specific characteristics of each of these, but our prokaryotic cells are single cells, and they are organisms such as bacteria. Our eukaryotic cells have organelles, so little organs within their cells that perform specific functions. These organisms can be single-celled or multi-celled. Any living thing that you can see with the naked eye is going to be a eukaryotic organism. All those prokaryotic things like bacteria are going to be microscopic. Organisms that are eukaryotic are plants, animals, fungi like mushrooms, and protists. So we'll talk about all of those groups here in a minute. All right, so all cells have a few things in common. They all share a set of characteristics, whether they are prokaryotic cells or eukaryotic cells. All cells are bound by a plasma membrane. So we talked about phospholipids in Chapter 3. Phospholipids make up the plasma membrane by forming a bilayer. And that plasma membrane works to isolate the cell from the surrounding environment. So it sort of defines the boundaries of the cell. All cells inside their membrane have a cytoplasm and cytosol. So a cytoplasm is basically like a cellular jelly. It's kind of like a fluid inside the cell that acts like air in a balloon. It helps to kind of inflate the cell, and it also holds whatever structures happen to be in there. The cytosol is kind of similar to the cytoplasm. They can kind of be used interchangeably, but cytosol is basically all the contents of the cell. So it's the cytoplasm plus anything that happens to be in the cytoplasm, whatever that may be. It might be organelles, it might be DNA, it might be ribosomes. All cells also have chromosomes. So chromosomes are our genetic material. It's our DNA. All cells have to have some instructions for life. And then all cells have ribosomes. So ribosomes are structures that make proteins. So whether you are prokaryote or eukaryote, you have genetic material and you have ribosomes to use that genetic material and produce proteins. And all of that is contained within the plasma membrane and supported within the cytosol and cytoplasm. So let's talk about our prokaryotic cells. So prokaryotic cells do not have organelles. They don't have specific isolated regions for their functions. But the same functions, energy conversion, energy acquisition, producing proteins, all of those things that are important for living organisms still happen in organelles. They just happen in the cytoplasm rather than isolated organelles within the cell. So the cytoplasm contains regions where those specific functions take place. So you might have an organelle in a eukaryotic cell that's sole purpose is energy conversion. Well, energy conversion still happens in a prokaryotic cell. It just isn't bound in a particular region, or isolated, I should say. But cells are still performing those same activities. So down here at the bottom, we have an electron micrograph taken using an electron microscope that we learned about in lab. We don't use those here at SWIC. They're quite expensive. But this is a bacterial cell. Along the outside, you've got a plasma membrane surrounded by a cell wall. And then inside you have your cytosol. So all of your cytoplasm with everything suspended in it. Inside you have ribosomes that can't necessarily see. They're there, you can't really see them. They are just floating around in the cytoplasm. And then you have this region not bound by a membrane of DNA, and that is called the nucleoid. It's just a region where your DNA is sort of concentrated. So on the left here, we have a cartoon example of this. You can see some other structures we're not really going to worry about too much in this class. I just want you to know that prokaryotic cells have all of those requirements for being a cell. Plasma membrane, the DNA, the ribosome. And then some specific characteristics to those prokaryotic cells as well. Prokaryotic cells make up 2 of our 3 big domains. So a domain is basically a large group, a big, huge group of organisms. There are 3 domains. We've got bacteria. You are all familiar with those. Things like E. coli and streptococcus. We often think of bacteria in a negative light. They're things that make us sick. They're dirty. They're not all bad. But one of our big domains is bacteria, and our second is Archaea. These are just really, really big inclusive groups of organisms. Those are our 2 prokaryotic cell domains. So bacteria, as I said, those are things that you are familiar with. They are single-celled prokaryotes, which means that they don't have any organelles, but they still have the plasma membrane and the DNA. Bacteria are found in every single environment, almost every single environment that you can think of on Earth. They're in our intestines, they're on our hands, they're in the dirt, they're on our skin, they're on rocks. Everywhere that something could live, there's bacteria living. And bacteria really are not bad. They're really kind of our microscopic friends. They do a lot of things for us. They just get a really bad rap. For example, you have a lot of bacteria that live in your gut that help process all that food you eat, that help make vitamins for you. They break down food for other organisms. They break down waste, dead materials, things like that. So bacteria are actually really, really important. Our other big group of organisms that are prokaryotic are Archaea. Archaea are probably less familiar to you. They are similar to bacteria in that they are single-celled prokaryotes, but they tend to be found in extreme environments. And you have maybe not ever heard of Archaea before today. They're found in places like deep sea sulfur vents. Super, super salty lakes. They are really great at adapting to extreme conditions. So that brings us to eukaryotic cells. So our two big types of cells are prokaryotes, and our organisms are bacteria and archaea. And then we have our eukaryotic cells. Eukaryotic cells are the cells that make up humans, and they make up your dog or your cat, that big tree that you can see out your window. All of those are eukaryotic organisms. So before we talk about the characteristics of eukaryotes, I want to take a little pause and talk about evolution a little bit. So life started basically as bacteria. And from that evolutionary line, we got Archaea, but those bacteria continued on. So these are both our prokaryotic organisms, but eukaryotic cells actually evolved from a special relationship that formed between Archaea and bacteria. And we'll talk about this, but Archaea eventually evolved into eukaryotes. Now most of those Archaea continued on in their own line. So we get these three big domains: bacteria, archaea, and eukaryotes. So eukaryotes are actually more similar to archaea than they are to bacteria, and that has to do with the way that eukaryotic cells evolved. We'll talk about that later in the lecture. So eukaryotic cells are made up of organelles. They are local environments within a cell that are enclosed by a plasma membrane, and they have very specific functions. So you can think of these like the organs in your body, where your heart has a specific function, and your liver has a specific function, your intestine has a specific function. Each of the organelles within our cells has a specific function. Our textbook is going to focus on the animal cell and the plant cell. Most of the organelles are the same. The same organelle has the same function no matter what cell it's in. But each type of cell has its own special characteristics. So some cells have organelles that others don't. We are going to focus on plant and animal cells, and we're going to go through and talk about the function of almost all of these cells. I do want you to be able to identify some of the characteristics of these cells though. Animal cells tend to be sort of rounded in shape. They do not have a cell wall, which means they're basically soft. They're not rigid. Plant cells, on the other hand, have a plasma membrane that is surrounded by a rigid cell wall. And that cell wall is what helps give it structure. Animals get structure from their bones. Plants and plant cells get structure from their cell walls. All right, so our eukaryotic Organisms are organisms that are made up of eukaryotic cells. And as I said a few minutes ago, these are all the things that you can see with your naked eye. Animals, plants, fungus. We won't talk too much about the fungus group, but they tend to have some characteristics that are similar to plants. Sometimes they're more similar to animals, but they are still eukaryotes. We just won't talk about them, really. We're going to focus on plants and animals. The eukaryotic domain is essentially broken up by how the organism obtains its food. So animals, you all know what animals are, they obtain their food by eating it. So consuming another living organism. The plant domain gets their food and energy from sunlight. Sorry about that. I'm not sure. I'm having some technical difficulties. So plants absorb sunlight energy and then they convert it into a usable form. The fungi kingdom, or the fungi group, gets their nutrients by absorption. So they absorb it from the soil around them. The protists are kind of a special group. They're not actually their own branch or their own group within eukarya. Protists are single-celled eukaryotes that belong to all three of these other domains. So you might have something like, is pictured here, this sort of blob here, is a paramecium. That is a single-celled organism that's kind of like an animal. It's basically an animal cell, but it engulfs, it swims around the water and engulfs its food. It's just a large single eukaryotic cell. Or you've got things like algae that are very similar to plants, that might be actually part of the plant kingdom. So protists are just kind of a broad category for anything that falls in any of these other domains that are single-celled eukaryotes.